Welcome everyone. Let me present myself, Priyanka Kande-Patil, Assistant Professor, Department of Microbiology. So today we will explore the microbial enzyme production and our main focus on the extraction, purification and industrial application of amylase enzyme. Started with basic introduction of microbial enzyme. Microbial enzymes are the biological catalyst for the biochemical reaction leading to microbial growth and respiration, as well as the formation of fermentation product or the desired product. So we know about the biocatalyst. Biocatalysts are nothing but the biological catalyst, which are the chemical substances responsible to accelerate the chemical reaction within the living organism. Biocatalysts, they are highly specific in nature. Each biocatalyst target specific reactant or substrate within the living organisms, and they are fitting together like lock and key. They are reusable. That means we use repeatedly over time. This particular biocatalysts are sensitive to certain environmental conditions. So we know that all enzymes are proteins, but all proteins are not enzymes. So enzymes are proteins, that's why they work under based optimum conditions. For example, normal body temperature and pH. If environment become too acidic or too hot, so this particular proteins lose their shape and stop working. Simply they undergo denaturation process. So this is the basic introduction of microbial enzyme. So by using various types of microorganisms and by utilizing different biotechnology tools, we can develop or we can form the desired microbial enzymes in less time or at a laboratory level. So moving forward, the microorganisms that produce enzymes. So the first category, that is bacteria and actinomycetes. So this particular category include Azospirillum, next Bacillus subtilis, Pseudomonas, Rhizobium, and Streptomyces. Next category, that is fungi, yeast and mold. So this particular category include Aspergillus niger, Aspergillus oryzae, Penicillium notatum, Penicillium chrysogenum, Trichoderma, and the most common yeast, that is Saccharomyces cerevisiae. And the last category, that is algae, which includes microalgae and chlorella. So these various types of microorganisms are responsible to produce microbial enzymes, including amylase, protease and lipase. Moving forward, so what is the culture media? Culture media, it is the source of nutrition during the production of enzymes for the microorganisms. So they can grow very well. Culture medium is nothing but the ideal medium which contain cheap source of carbon, nitrogen, amino acids, trace elements and little amount of salts within it. So this particular culture medium is responsible to support the growth of microorganisms at the certain laboratory conditions to obtain a maximum amount of microbial enzymes. So two types of cultures we can use in this enzyme production process. The first culture, that is semi-solid culture, and second one that is submerged culture. So the semi-solid culture, it is prepared with agar at concentration of 0.5% or less than 0.5%. It has soft custard-like consistency. So here we use a semi-solid substrates. For example, moistened wheat and rice bran with lots of nutrients in it, and which is responsible for the growth of microorganisms, and this particular microorganisms give the maximum amount of enzymes. The second culture that is submerged culture. So here instead of semi-solid substrate we can use liquid media with lots of nutrients for the growth of microorganisms. So moving forward, let's discuss about the types of amylase. There are three types of amylase. The first type that is alpha amylase, which is the endoenzyme. So what is the endoenzyme? It is also known as intracellular enzyme. So this particular enzyme function or secrete inside the cell. That's why it is called as endoenzyme. So this particular alpha amylase responsible to cleave or break the internal alpha-1,4 glycosidic linkage. It is also known as alpha-1,4 D-glucane glucanohydrolase. So this particular alpha amylase acts as the calcium metalloenzyme. The calcium ion strictly required for the enzyme catalytic activity, thermal stability, and structural integrity of that particular enzyme structure. And this particular type of amylase work under the pH range that is 6.7 to 7. Approximately the neutral pH range is required for the growth of that particular alpha amylase enzyme or the production of that particular alpha amylase enzyme. And this particular alpha amylase is responsible for the breakdown of starch into two units. First, that is amylose, and second one is amylopectin. So amylose and amylopectin, these two are the primary polysaccharides. The structure of amylose include linear unbranched chain, and amylopectin include highly branched chain. This particular amylose responsible to cleave or break alpha-1,4 glycosidic linkage, and amylopectin, this particular unit responsible to cleave both alpha-1,4 and alpha-1,6 glycosidic linkage. The amylose is 20 to 30% proportion of that particular amylose present in starch, and typically 70 to 80% proportion of amylopectin present in starch. So amylose have slower digestive capacity, and the amylopectin has rapid digestive capacity. Next type, that is beta amylase, which is the exoenzyme. Exoenzyme, which is also known as extracellular enzyme. This particular type of amylase work or secreted or function outside the cell. That's why it is called as exoenzyme. So it is also known as alpha-1,4 D-glucane maltohydrolase. Why it is called as maltohydrolase? Because the non-reducing end producing maltose. That's why it is called as maltohydrolase. This particular beta amylase enzyme synthesized by using different types of microorganisms, then fungi and plants. So this beta amylase, during the ripening of fruit, this beta amylase is responsible to break the starch into maltose sugar, and this maltose sugar give the sweet flavor to that particular ripe fruit. That is the function of beta amylase enzyme. This particular type of amylase work under the pH range that is 4 to 5, that is acidic pH range. The last type, that is gamma amylase, which is also known as glucoamylase. Why it is called as glucoamylase? Because This particular gamma amylase is responsible to break the amylose and amylopectin into glucose. That's why it is called as glucoamylase. So this particular gamma amylase is responsible to break down both alpha 1,4 as well as alpha 1,6 glycosidic linkage. And it is also known as alpha 1,4 glucosidase. So this is all about the classes of amylase enzyme. Moving forward, the microbial producers of amylase. It includes bacteria, fungi, and yeast. Bacteria, for example, Bacillus subtilis, Bacillus licheniformis, and Bacillus amyloliquefaciens. So basically, Bacillus amyloliquefaciens, it is the primary industrial producer with a high thermostability. Next category, that is fungi, which includes Aspergillus niger, Aspergillus oryzae, Penicillium notatum, Penicillium chrysogenum, and Rhizopus species, with excellent for the acid stable amylases. So these all are acid stable amylases. The third category, that is yeast, the most common yeast, that is Saccharomyces cerevisiae, and the Candida species, which are responsible to produce the amylase enzyme. So why these organisms prefer to produce the amylase enzyme? Because they have generally recognized as a safe status. They are high extracellular enzyme secretions, well characterized genetics, stable fermentation profiles, and they have high thermostability. That's why we prefer these microorganisms for the amylase production. Moving forward, step by step amylase production. So here the first step, that is strand selection. Strand selection is nothing but the selection of microorganisms. So here microorganisms preferred which produce maximum amount of enzymes in less time. So for this strand development, here we can use different types of mutagens. Strand development or strand improvement, it is the process where we modify the genetic makeup of that particular new industrial microorganism to enhance the desired properties of that particular microorganism. So for this strand development, we can use horizontal gene transfer techniques including conjugation, transformation, and transduction. Also, we use classical methods including UV light and chemical mutagenesis. So in this process, we can expose the cell to the UV light and certain specific chemical agents that basically change their genetic makeup. Also, we use the targeted approach technique, that is CRISPR-Cas9. So CRISPR-Cas9 system, it basically clustered regularly interspaced short palindromic repeats with associated with Cas9 protein. So this particular system is responsible to find, delete, and replace the faulty section of DNA, and it is responsible to cure the genetic disorders as well as enhance the crop yield and recover the illness, for example cancer. So this strain improvement techniques we use to develop this strand, which give the maximum amount of enzyme. Next step, that is inoculum preparation. So the pure culture of selected microorganism is maintained on the nutrient agar slants as well as starch agar slants, and they are particularly transferred to the liquid medium as the inoculum. Next step, that is media formulation or the preparation of fermentation media. So this particular medium contain soluble starch, peptone, yeast extract, mineral salts including KH2PO4, then magnesium sulfate, calcium chloride, Na2HPO4, potassium chloride, and magnesium chloride. We maintain pH range that is 6.8. Some key nutrient sources also used in the media composition including carbon sources including maltose, sucrose and glucose, nitrogen sources including ammonium sulfate, sodium hydrogen phosphate and ammonium chloride. And some common substrates are used here, that is molasses, corn stip liquor, peptone, yeast extract, malt extract, soya meal and whey. Moving forward, next step that is the fermentation process. Here we use two fermentation techniques. First is submerged fermentation and second one is solid state fermentation. So in submerged fermentation technique, microorganisms grown in liquid medium containing starch, peptone and yeast extract. So this particular submerged fermentation technique is used to produce the bacterial amylase enzyme. Next, solid state fermentation. So here we use the solid substrates like wheat bran, rice bran, cassava. So these solid substrates are used to grow the microorganisms, and this particular fermentation technique used to produce the fungal amylase enzyme. So here some optimum conditions are required. So for the bacteria, temperature is maintained 40 to 45 degrees Celsius or 55 degrees Celsius, pH that is 6 to 7. And for the fungi, temperature is maintained 30 to 40 degrees Celsius, pH range that is acidic 4.5 to 6. So here the aerobic condition is maintained. This whole procedure is occurred in the presence of oxygen, and we require the production time that is 3 to 5 days. So within 3 to 5 days, we obtain a desired product that is amylase. Moving forward, let's discuss about the recovery and purification of amylase enzyme. So here the fermentation broth is filtered and centrifuged. So this particular process, that is filtration and centrifugation, is responsible to remove cell debris, certain impurities and solid substrates which are present in that particular fermentation broth. That particular process is known as clarification. So next, there is the removal of nucleic acids by adding polycatons, include streptomycin, polyamines and polyethylamines. That particular polycatons are responsible to precipitate that nucleic acid. Next step, that is precipitation by using ammonium sulfate and ammonium hydroxide. These particular chemicals are responsible to precipitate amylase, and this precipitate is collected by using filtration technique. We use here filters. Next, the purification. So we purify that particular desired product by using the chromatography techniques including gel filtration and ion exchange chromatography. Along with that, we use dialysis. And lastly, the crystallization and drying. The precipitate is crystallized at 4 degrees Celsius, and then this precipitate is dried by using film evaporator and freeze dryer. So drying is required to remove the moisture content present in that particular desired product to avoid the contamination. So this is all about the production steps. Last part, that is application of amylase enzyme. We know that amylase enzyme is widely used industrial enzyme in food, fermentation technique, textile, detergent industry, medicine, pharmaceuticals, etc. So first we say food and beverage industry. So here amylase enzyme is used in baking industry. So particularly amylase is responsible to break the starch into fermentable sugars. And this particular fermentable sugar is responsible to improve or to handle the texture of that particular bread. Next, that is brewing and distilling industry. So in this particular industry, the amylase enzyme is used in beer and whiskey production, and it is converted into cereal starch, into the maltose sugar. Also, it is converted into glucose for the yeast fermentation process. Then it is also used in fruit and juice industry for the clarification purpose, that basically increases the sugar content to enhance the flavor of that particular fruit or juices. Next, application in textile and detergent industry. In textile, it is used in desizing of fabrics, and in detergent industry, the amylase enzyme is responsible to remove the starch-based stains. Food stains basically include rice, gravy, chocolate, and custard from the cloths. Next application, that is paper and pulp industry. So amylase enzyme used for the starch modification in paper coating, then improve the strength of that particular paper, then smoothness and the print quality of that particular paper. Next application in pharmaceuticals and medicine. So amylase enzyme act as the digestive aid. Basically, the amylase enzyme play important role to diagnose the pancreatitis. So pancreatitis, this particular disease, is nothing but the inflammation occurring in pancreas. Next application, biofuel industry. Amylase enzyme play key role in bioethanol production and dairy industry. So amylase enzyme is used to produce the lactose-free and low viscosity dairy products to avoid the lactose intolerance in humans. And the last application, that is waste management by using various types of microorganisms, and by utilizing different biotechnology tools, we can easily develop or we can easily produce amylase enzyme in laboratory level in less time. So this is all about the applications of amylases. So thank you so much. This is all about the microbial production of amylase enzyme.